So today I'd like to start off by talking about the work that's been done in culture-based methods to understand the role of bacteria in HS, and then go on to talk about how these findings can be extended using sequenced-based methods, and then present some data both from the literature and some of our own that lend some insight into the microbiome of HS. So these are my conflicts of interest. I'm a board member of the HS Foundation. I'm also a co-investigator on a research grant that's funded by AbbVie. So despite the clinical presentation of hidradenitis suppurativa, including inflammatory nodules, abscesses, and chronic suppurative and malodorous drainage, we don't believe that HS is an infectious disease, and I know there's a debate coming up a little later this morning, so we can kind of get into that a little bit later. But I think we can all agree that bacteria does seem to play some role in HS, and today I want to talk about the efforts that have gone into understanding what that role might be. So in this systematic review by Ring and Dr. Yemick and colleagues, they looked at 9 studies that examined biopsy specimens for bacterial growth in the setting of pure culture. And what they found in the over 300 specimens that they looked at was that 50% of the lesional specimens were actually culture negative. And in the other 50%, they found that the organisms that predominated were typically coag-negative Staphylococcus and anaerobes, and Staph aureus, shown here in royal blue, and Corynebacterium, shown in light blue, were also made up a significant proportion of the organisms that grew in culture. These findings have largely guided the use and the choice of antibiotics for the management of hidradenitis suppurativa. But we know that antibiotic therapy, even combinations of antibiotics, are really kind of modestly effective in the management of HS, and where they are effective, they seem to be most effective for the patients who have more mild disease. We have in recent years made great strides in trying to offer patients other better options for treatment for the management of their hidradenitis. And specifically, I'm talking about biologic therapy, particularly with TNF-alpha blockade. And the patient's responses, disease response to TNF-alpha blockade, have really pointed to the important role that immune dysregulation plays in hidradenitis suppurativa. So how do we begin to make sense of both of these pieces of information, that we do find bacteria in subsets of lesional specimens, at least based on cultures, in HS patients, and that these patients clearly have evidence of immune dysregulation? So one way that we can make sense of that is if dysbiosis, or abnormal pathogenic microbial colonization, plays a role in hidradenitis suppurativa. So the skin environment and all of the microorganisms that reside in and on the skin make up what's known as the skin microbiome. And we know that in other inflammatory diseases, alterations in the number of bacteria, the types of bacteria, the proportions of bacteria can lead to inflammatory disease states, or can contribute to inflammatory disease states, and that has been shown in data in atopic dermatitis and inflammatory bowel disease, and to some extent in psoriasis as well. Now, less than 1% of bacterial species can be cultivated under standard laboratory conditions, so using culture-based methods is not going to be the best way to assess all the microbes that live on the skin, that reside on the skin. And so instead, culture-free or sequence-based methods really are the way forward in terms of trying to take a look at a comprehensive and unbiased look at what microbes exist on the skin and what microbial shifts may be occurring in the setting of inflammatory diseases. And really being able to sequence the DNA of microbes gives us a better picture of the spectrum of microbes that exist on the skin without having to take into consideration whether those microbes are cultivatable in pure culture. So using those sequence-based methods, we have learned that bacteria, fungi, viruses, and mites colonize the human skin, and commensal bacteria make up a large proportion of the organisms, microorganisms that live on the skin surface. And in fact, commensal bacteria serve as the body's first line of defense against pathogens, and the cutaneous immune system, which is kind of in the background and surveying and recognizing and making distinctions between commensal organisms and pathogens, really is the second line of defense. So I want to talk a little bit now about the data, the body of evidence that tells us a little bit about the microbiome in HS, and there are only a couple of studies that have been reported to date, so the body of evidence is small but growing. This is a study that looked at 1 abscess and 5 active HS lesions in HS patients, and this was essentially a proof of concept study to prove that we could, in fact, use sequence-based methods to understand the proportions and the types of microorganisms that exist in Hidradenitis suppurativa. The abscess, the single abscess that was sequenced, is the bar on your left, which showed almost entirely a huge proportion of Staph lugdunensis, and then the remaining 5 bars to the right of that green bar represent the proportions of bacteria that were seen in the 5 additional active cutaneous lesions of HS that were biopsied. And what you see here is that there's a huge proportion of anaerobes, as shown by the red and the orange portions of those bars, and the proportions of coag-negative staph, of Staph aureus, and of Corynebacterium is far less than what the culture data that I initially showed you from the systematic review would have led us to believe. So earlier this year, Dr. Yemek and Dr. Ring and colleagues published this paper looking at the follicular skin microbiome in HS patients, and this was really a more systematic look at the skin microbiome in patients with hidradenitis suppurativa. And they looked at both lesional and non-lesional skin biopsy specimens from the axilla and the groin of 30 patients with hidradenitis suppurativa. And they also compared these data to 24 specimens from the axilla from healthy volunteers. And they used 16S ribosomal RNA sequencing to sequence the specimens that they obtained. And in this diagram, let me orient you. So on the y-axis is the relative abundance, the mean relative abundance of microorganisms, of bacteria that were found in these specimens. And then on the x-axis, the first third of the x-axis represents lesional skin. The middle portion here is HS non-lesional skin, and on the last portion here is healthy controls. And what you can see just at your initial glance is that all 3 of these blocks, based on the colors here, which represent different bacterial species, all 3 of these blocks look quite different. So non-lesional skin in HS patients has a different microbial composition than healthy controls, and certainly a different microbial composition than lesional skin. And I think that's one important takeaway from these data. What you'll also see is that there seems to be a significant proportion of anaerobes in the patients, in the HS lesional skin specimens, as well as Corynebacterium and Firmicute species from the Firmicutes phylums in the HS lesional skin. In the HS non-lesional skin, Acinetobacter species and Moraxilla species really predominated, and then in the healthy controls, as we might expect, they saw a lot of higher relative proportion of P. acnes and Staph epi, which were not seen in the other two groups. When they looked at the richness of these groups, which essentially is the number of species in each of the specimens collected, they did not find any significant differences in the richness between HS lesional skin, HS non-lesional skin, and healthy controls. But then when they looked at the Shannon Diversity Index, which is a measure of the both the richness and the evenness of species in these specimens, they found that actually the HS non-lesional skin specimens had increased evenness, and therefore they had an increased Shannon Diversity Index as compared with HS lesional skin and healthy controls. And the reason that this is interesting is because we've seen in other inflammatory diseases that actually the Shannon Diversity Index in the setting of inflammation tends to be lower than what we would see in the setting of baseline disease or kind of quiescent disease, and certainly lower than the Shannon Diversity Index that's observed in healthy controls. So there's still, I think, quite a bit to learn about what is going on in terms of the microbial shifts in Hidradenitis suppurativa and why we see these differences in community richness and community evenness in the Shannon Diversity Index. So I just want to, if I may, present a little bit of the data that, of some of our initial data that we've collected, which corroborates the findings in Dr. Yemek's study and also, I think, begins to extend those findings as well. So we looked not only at the axillary and groin specimens, so moist body sites, but we also looked at sites of HS predilection, so more atypical sites like the back, the chest, and the retroauricular crease. And what we found was in agreement with Dr. Yemek's work, which is that we found increased proportions of anaerobes as well as Corynebacterium in the moist sites, so the inguinal crease and the axillary vault of these patients. And at these more atypical sites, we found that these sites were notable for increased proportions of staph species, Firmicutes species, and also Corynebacterium. So what we're seeing here compared to healthy volunteers is that in both HS-affected skin as well as in sites of predilection, essentially there are significant shifts, microbial shifts in the microbiome of these patients. And so the skin that we're seeing, even when we're not seeing disease, is not totally normal. So, excuse me. So in summary, based on kind of these initial data, I think we can safely say that we are seeing evidence of alterations in the skin microbiome of HS patients as compared with healthy volunteers. There seems to be a relative abundance of Corynebacterium, Firmicutes, and anaerobes at HS-affected sites and at sites of HS predilection. And sequence-based methods can provide unbiased and comprehensive assessments and insights of microbial shifts that may be relevant to HS. And then the next step, once we kind of have a better sense of what these shifts are, is trying to understand what those shifts really mean in terms of the host response that's elicited. So stay tuned, there's more to come. Thanks very much.